This is a production of Cornell University.
I'm Michael Mazourek. I'm the vegetable breeder, tending to focus a lot on peppers, cucurbits, and peas, and so that's what we're going to hear about today. So the title of my talk is The Major Genes Affecting Vegetable Quality Traits via Phenopropanoid-derived Metabolites. So one of the things that I'm particularly interested in is in chemistry, and here are some of the pictures related to that. A cool peat that my student James is working on that is having some anthocyanins there from the phenylpropanoid pathway. And then a couple other really cool pictures I just got back from an undergraduate student that is in Austria now, that is sending me back pictures of all the different flavorful paprikas that she's encountering, and also all of the pumpkin seed oils. So this is actually what we're going to talk about today. And so for me, the phenylpropanoid pathway has been something that unifies a lot of my interests in vegetable quality. So a series of compounds that come from phenylalanine are converted into the phenylpropanoids, so phenyl for the phenyl group and propane for this 3-carbon group here. And then they go off to do lots of useful functions within the plant. A lot of them are involved in disease resistance. In the work I'll talk a little bit about today and have talked about a lot before, capsaicin, what makes hot peppers taste hot, into all the cell wall fortifying compounds for lignin production. And then also off into the colorful antioxidant anthocyanins. And along with that pathway, I'll also talk a little bit about another group of compounds, the proanthocyanins, that in many foods can have a very nice addition to flavor, but in some of the examples I'll talk about today, actually they are one of the major detractors from flavor. And so this is kind of why I tend to focus on this pathway. And a lot of what we do about quality is going to come from and center on the metabolites in that pathway. A lot of what I had been doing the first couple years here is focusing on disease resistance. So the two major threats that we've encountered that growers have asked us to work on is here on pepper suffering from Phytophthora blight, and here is a cucumber leaf that's suffering from downy mildew, another oomycete pathogen. And one of the first things I've really worked on is finding some sources of disease resistance to these major diseases that we had as breeding lines in the program, and starting to do some more work just evaluating them. And so here is an example of a pepper that is doing fine despite all of the many spores of Phytophthora capsicii that Chris and Amar have inoculated it with. So it is doing much better than that pepper there. So we have some excellent sources of disease resistance for Phytophthora here. And for downy mildew, while it will cause cucumbers to typically and quickly look like this in the field in late summer, having some sources of resistance, just going through the collection, seeing what we had, we were able to find that we do have some significant sources of downy mildew resistance. So this, our first inquiry into what we had in the program already. Could we have varieties that could combat these diseases? Yes. But so then, since these are some major grower needs that they continue to voice, looking through the program where we had solutions to this, we had some big questions of, so why aren't these breeding lines or these varieties used more broadly? One big issue turned out to be definitely grower awareness, that we had these resources. Luckily, I was able to connect with Chris Smart from Geneva early on in the work here, and got some varieties up to the Phytophthora farm in Geneva. They turned out to be quite resistant. We had some really promising material. And so then the next step, and something that Chris helped me orchestrate, is really just connecting with all of the great extension personnel around the state. And right now, I'm happy to say that a lot of our seed from those resistant peppers, as a result of the collaboration with Chris and now working with the much larger group of folks, is actually getting out onto growers' fields. They're starting to sow the seeds this week. So one big hurdle is overcome there. So now we have these resistant varieties that growers are starting to learn more about. The other question, though, that why they might not be using these varieties is a question of yield. So here is one of the results from our Towards Sustainability project from last summer, where we're looking at yield of cucumber plants planted in a late season planting out in the field on the organic plots in Freeville, where we're looking at the weeks after transplant along this axis, and the cumulative weight we harvested off the plots on that axis. And what we saw was that although we had some plants that quickly, just almost spontaneously combusted in the field, that would be totally decimated by disease, and some of our resistant lines we kept trucking along and still looked quite disease resistant, it was actually some of these highly susceptible lines that had the most yield. So two popular lines here, Dasher 2 with the triangle, and Piccolino, the variety actually pictured here in the open squares, they were highly productive despite being highly susceptible to disease. A lot of our resistant cultivars down here, so for instance, Markerassisted 97, which is commercially available and turns out to be highly resistant to downy mildew, just really doesn't quite have the yield to compete with the other ones. And as a grower, you sell cucumbers, not cucumber foliage. So there's a big need to, from the grower's perspective, they want resistant varieties, but they also need to have the yield. So in this case, it's not a lost cause. After looking at doing the formal comparison last summer, the clear difference between this group and this group is whether or not the crop is gynoecious. So having an all-female crop, you can see if we take Markerassisted 97 and just simply insert the genetics for all-female flowering, we should be able to quickly get up to this level of yield, if not higher. So looking at what we had, we know for some of these yield is a problem, but and that's the reason why growers would be using it, but we know how to overcome that. So that's probably also not going to be an issue. So the third reason, something that we are looking at in our varieties to actually make them a complete package, something that the growers will really want to use, comes down to a matter of quality. And that's what I came to talk today about, and it's a good time because now that we're starting to really refocus on quality in my program and the chemistry behind that. So it's been a hypothesis in the literature for a while. It really became popular when Harry Klee was proposing it a couple years ago, where he was doing surveys of all the aromatic volatiles in a vegetable, everything that we think smells really good about a fruit, and finding that those aroma volatiles we like tend to be derived from byproducts of essential nutrients produced in the plant. And other people have also talked about the same phenomenon, and it's the notion that a lot of the sensory cues we get from a plant is really cueing us into its nutritional value. So all of the colors and aromas we might get from different fruits and vegetables are really what's cueing us in as to these are really healthful and good. And the story that Harry was talking about, and is kind of a lot of people are familiar with now, is taking a look at either the grocery store tomato that's become a bit of a cliche now for being pink and crunchy and flavorless, and then some of the much more flavorful, much more pigmented, lush cultivars that can be obtained, or even some of the novel niche categories as people are starting to more and more want to try to paint vegetables purple. It's a common theme in a lot of plant breeding now, is to have a purple crop. So, you know, again, the idea is with the anthocyanins, there's an extra health benefit. And so even in asparagus, there's the purple asparagus available. One thing here is kind of against the hypothesis is our white asparagus here doesn't have the visual cues, but in terms of its aroma, it actually has more of some of the essential amino acids than the others. So it's always a balance between what can we produce that really fits in the mainstream views of quality, what the consumer wants in quality, and how much room is there for these niche vegetables that also are high quality but look a little bit different than some people expect. But there's been, like I mentioned, with painting some of the crops purple, there's been some great success. Just a few days ago, I was talking with Frank Morton, a lettuce breeder who is readily able to do his own plant breeding and selection on farm for different salad green mixes that have an array of different anthocyanin profiles, and along with that comes some very different interesting flavors. Over in England, there are some amateur plant breeders that are actually reintroducing us to some pea varieties that haven't been seen in a long time, in some cases recreating them. And so here is a woman that has a website to showcase her pea breeding talents, where she is trying to recreate a red potted snap pea. So the problem with working with some of the anthocyanins is that it's not just as simple as painting the fruit a different color or the pod a different color. So this is a quote, actually, I got from a Geneva student, Gary Tarek's thesis from the 80s, and we're just kind of rediscovering some of this work now. That although you can get these pretty colors in the flowers, and then along with that you tend to also get anthocyanins in seeds, and you can also have other transcription factors that will further turn on the anthocyanins in pods, having this anthocyanin upregulation is unfortunately associated with really poor quality. For some of the processors, that would be because when you go to canned purple peas, you end up with a can of gray, dingy gray peas. But in terms of the pod, it's actually, you get some flavor profiles that really aren't what you're after. And to jump back to the original slide, what the source of that is that when you turn on the anthocyanin pathway to have all these flavonoid colors in the pea, you also end up also upregulating the proanthocyanin pathway, condensed tannins, which might be really nice in a cup of coffee or a dark red wine, but are not necessarily the flavor profile you want to go for with a sweet sugar snap pea. It's kind of conflicting. So we wanted to start to do some work to genetically dissect what was possible in pea. Can we separate the two? Some things were already known, but we started to go after that with a new set of tools. So we started out, this is some work from my student, James Keach, where we started to get a collection of different pea flowers, so a typical garden white flowered pea, the yellowish orange flowered Pisum fulvum, a different species, also some pink flowered, and some purple flowered peas. And here's just hydrochloric acid and methanol extract of the different flower parts. And I should also mention that in starting to look at some of the connections between color and the proanthocyanins, it's also something that's of interest to the forage folks. And so to kind of broaden the study, started a collaboration with the other new hire in the department, Royce Murphy. And we have, and so we're starting to look at some of his alfalfa flower colors, where he has challenged me that any flower color I can find in a pea, he has the same thing and better in alfalfa. And so we're starting to integrate some of his breeding lines into the analysis also. But anyway, so just to look at a couple of these now, or actually let's look at the chemistry of it all. So as we start to do the analysis, one thing we can take advantage of is there's really only 6 major anthocyanins in nature. There's some other forms, but they're really quite rare in some more obscure species. But we have these 6 major forms that just vary on whether they're hydroxylated at some of these R group positions around the ring. But they can be very, very extensively diverse based on what sugar moiety they have acylated to these 2 positions. But since they all have some of the same core structure, we're able to take advantage of that in the analysis. And so here is a lot of chromatography that was done across the street by Raiko Halishi. And so what we did were to remind us we had the different pea flowers, that we had the methanol extract, and Raiko ran it through an HPLC where on the top graph we have a chromatogram of the photodiode array, so looking at the light absorbance, and having all different colored molecules show up here. And then within that, what he'd do is he'd cue in on all of the different core anthocyanin base molecules, so without all the sugars added. And so by having selected masses to pull out of here, he could then go, for instance, say that he knows the 287 mass corresponds to cyanidins, the 301 mass corresponds to peonidin, and could pull out what are all the different core aglycone structures of the anthocyanins. Then the nice feature of the particular type of mass spectrometer he has is then, once they've pulled out those masses and can line those masses up with a peak, they can go back and do what they call a precursor ion scan. And so basically ask, so what are all the molecules that when they broke down resulted in having the peonidin core group? And so then we can start to look at all of the different sugar moieties. So while we can just focus on the 6 major anthocyanin aglycones, Raiko is now looking at least 39 different anthocyanin groups that he is finding. So here is a pink pea flower showing the cyanidin and peonidin. And then here is the same experiment for this purple flower, where you can see the mix of delphinidin, petunidin, and melvin. And so this is not anything that wasn't necessarily known before about the pea flowers, but we're just able to start to look at it with a lot more detail and accuracy, and to start to really explore and find actually new compounds that weren't reported before. And then, the end goal will be to, within the same spectrum, to try to be able to capture all the flavonols, the quercetins that are important health compounds, and also some of the proanthocyanidins, which are the bittering compounds in the peas. And this is, of course, it's very interesting for us to see what are all the different anthocyanin compounds in the peas, and study a little bit more about the molecular genetics behind it. But also, as part of a larger breeding project in collaboration with Jim Myers, is to start to develop some more stringless sugar snap peas for the area. And for me, to start to get some that have some of the purple coloration. And so again, here we're after the full package of having a purple stringless snap pea. So for stringlessness, that is a single recessive gene to remove the string. In the grocery store, most of the peas you'll see now, you'll notice they are missing the stem because they are starting to become more and more common to have the stringless snap peas because they're more readily machine harvestable. Then for the snap pea, it's actually a combination of 3 different recessive genes, 2 recessive genes that can combine to remove this inner parchment layer, and then a 3rd that's involved in making the pod swell to make the snap pea. So here would be the snow pea, and then there would be the snap. With the problem working with some of the stringless material is by virtue of it being stringless, we're starting to affect the phenylpropanoid pathway. We're reducing the plant's ability to produce some phenylpropanoid-related defense compounds. Some of the sclerenchyma tissues that are being removed also in the process, especially with the massively pleiotropic stringless allele. Having lots of other phenylpropanoids change within the plant. And so we have a struggle now to try to figure out how to balance a lot of different traits. And so, again, and one of the issues has been, how can we produce anthocyanins without producing the proanthocyanins? So there are some genes that have been reported that can actually decouple those, and so that is something we're looking at now. Recently, it's become available, actually a purple snap pea that is claimed to not have some of those bitter off flavors. So it's touted as the first good tasting purple pea. And so we're got some of that. I haven't tried it yet. We're planning it out for the first time this year. But looking at the seed coat of the peas, so this is the seed coat of a typical purple potted pea, and you can see some of the anthocyanin speckling on it. Whereas here we have a pea from the new variety, Sugar Magnolia, a great name for a purple pea, and it's lacking the purple speckling universally. One of the genes that is known to be able to remove some of the proanthocyanins would also cause this effect. So for us, it's going to be also interesting as we start to look at these genetics and some of the other genetics that are available to decouple these, is with the stringless mutation, as we're stopping the lignin production and affecting the rest of the phenylpropanoid pathway, you start to lose that cold soil germination, the early young seedling disease resistance. Whereas once you add that dominant A gene, you turn on anthocyanin biosynthesis, actually the seed coat becomes much more disease resistant. And so it's going to be a question of trying to figure out how the stringless genes interact with the anthocyanin genes to see how they are not only affecting quality and plant vigor, but also potentially that early seedling disease resistance. So right now, that is a work in progress. We'll be looking at some of the peas again this summer and continuing to look at some of the anthocyanins. The other project, this one is again focusing largely on some of the lignin and hemicellulose biosynthetic pathways. Starting to look at squash seed oil. So up in Geneva, Greg Woodworth at the Stony Brook Wholehearted Foods has started up a very great business pressing seeds that are left over from the squash processing into a really nutritious, flavorful oil. And so Greg is taking advantage of something that's known in Austria and has been known for quite a while. Is in Austria, the pumpkin seed oil is a very desirable flavor. Here's an image Leah sent me back of some pumpkin seed oil flavored chips. This lady here is not bemoaning the grass stain on her skirt, but rather just being, I guess, as part of the pumpkin seed oil ad, being excited that she is part of Austrian culture by getting some of the pumpkin seed spilled on her. This is the actual advertising for the oilseed company is it stains. It does stain quite a bit. You can see, so here are some of those mutant seeds that are used for the oilseed production. And they're very high yielding because they lack seed coats. And as a result of lacking the seed coats, so the inner layers are more upregulated, and so you have a really dark green layer surrounding the seed, and that is what gives the stains on the clothes. So we've started to do some work into the genetics of this trait as well, as it ties in with understanding how the phenylpropanoid pathway is affecting some of the quality. So here on the far side of the slide is an example of some different hulless seeds that are available. So down here on the bottom are some of the commercial oilseed pumpkin seeds. And up here are some others that are, I don't think is used widely in production, but still have the hulless trait. So here would be like Kakai, and here would be a Lady Godiva. So that's some of the naked squash seed, or the hulless ones. And here again in cross-section you can see where there's really no seed coat. Some of these have a little bit along the edge of kind of the lignocellulose strip. But you can really see the difference as you start to look in this column and how radically the seed coat thickness can change as we go from a virtually hulless seed to a thin hull that's found on like Cucurbita pepo seeds. So here's a summer squash seed. Here is a Cucurbita moschata seed, which is starting to get more of a seed coat and tending to be quite rough. And then here, in the usually beautifully wood-grained looking Cucurbita maxima seeds, the seed is quite thick. And so what we wanted to start to do was to look at the seed coat in a little bit more detail, as we're going to try to study the gene in more detail, as a project my student Lindsay is working on, is to try to figure out, what more should we know about the seed coat. And so a great opportunity we found across the street. So over in Wild Hall, they have a CT scanner with some quite good resolution. And so what we started to do is try to see, can this help us with phenotyping the seed, or at least help us to see things we were missing before in the seed. So here's a 25 micron resolution scan of the Cucurbita maxima seed that in the slide before you can see has this really thick seed coat here. And we can actually go in and measure it at different points along the seed coat to see, different, in this slice it's ranging from 782 to 567 microns. And compare that to a Cucurbita pepo seed, which is much thinner, and so we can actually start to really get some nice measurements of it. Then beyond just getting one static slice through the image, one thing that we've been able to do is start to look at this in 3 dimensions. And so for the folks in Geneva, I should point out that I'm going to hit play on the 3D seed movie now. But to start to do some of the 3D rendering capacities of the CT scanner, so here is at Success PM, the Cucurbita pepo summer squash seed. And by playing with the ability of the X-ray to see different densities, we can dissolve away the seed coat, get a good look at the embryo, where the cotyledons are the major oil storage tissue, and look at it in various dimensions, and also then be able to go through the seed and be able to measure basically at any depth angle we want and see how the embryo is actually filling it as compared to where we see the seed coat. And then back to the slides. And so, as we're starting to look at the seeds in more detail, one of the reasons that's going to be important is there can be degrees of the seed coat present. And so one thing we want to do in being able to not only get a good handle on what are the different qualitative traits, there's one qualitative gene that's modifying whether or not we have the seed coat, but there's also some quantitative traits that can cause different degrees of the seed coat to appear. And to get a truly naked-seeded, hulless cultivar, we really have to do a better job of understanding what all these modifiers are so we can use a marker-assisted breeding strategy. So part of what we're doing is just crossing also this naked-seeded, hulless trait into a delicata squash, and you can see there's varying degrees of delicatiness emerging from some of those breeding lines. And also introducing it into an acorn squash. And here, the goal is to have a squash that, with the processing squash, they have a very nice system now. So after they make in the grocery store, you have the cubed, peeled, ready to cook squash. That's done at a plant. They can, if they want to press the oil for seed, they have the facilities and the setup to do it. But for the person at home, if you just get an acorn squash and cut it open. The seeds are very healthful, very nutritious. I think they taste great. It's a question of, could we make it so when you open up the squash, you get those same, naked seeded, oil seed, high nutrient seeds. Other things we're doing also is, I know every time we grow zucchinis, we always end up with a couple huge lunkers left over at the end of the season that just go straight to the compost bin. This is also a trait we're inserting into zucchini so that all those leftover squashes at the end of the year we can have a nice, nutritious second crop out of, essentially. And one of the tools we had hoped to use in this process was a number of linked SSR markers. There's tightly linked to the major hulless trait in squash. And so here we started to try to use a marker-assisted back cross strategy to be able to insert the hulless trait into zucchini. And we got some excellent results at first. And it turned out to be a really good thing that we kept all the plants and then just throw them away. It turns out that the SSR markers that we had started using turned out not to be linked to the trait. So I figured this would be my one chance to show all the great molecular marker work we did very intrepidly, but turned out to be it's very clear indication there needs to be some work done in actually getting some new markers for the trait, and to try to, definitely lots of room to actually now start moving towards a better molecular understanding of the nature of the gene behind the trait. And also, besides this being potentially interesting new cultivars, all of these different breeding lines are different introgression lines also be very important for trying to study the trait. And I think Lindsay will be telling you more about how the project's going in the coming years. So the last vignette I wanted to tell is about hot peppers and capsaicinoids. Some of you have probably heard a lot parts of the story before, but I really like the story, so I hope you do. One of my interests early on was in habanero pepper flavor, this hot pepper flavor, and how the hot pepper seemed to have flavors that just a regular pepper doesn't. Started to work with some spice companies that were also very interested in having pepper flavor, and how can you have pepper flavor without burning out your customers. And once McDonald's is starting to feature chipotle flavored, roasted red jalapeno flavor as one of their major menu ingredients, there's a big interest in the flavor, but how can you separate flavor from heat? Because hot pepper flavor, you always need to have hot pepper flavor in hot pepper. Different approaches have been tried. So this is a not hot jalapeno called Fool You. Sometimes you get one that has outcrossed, and then it's a whole different joke on the consumer. And then the habanero is the hottest, or was the hottest hot pepper, and is also for the people that are intrepid enough to try it, the source of some really interesting, wonderful flavors. And so what we wanted to start to try to do is develop a pepper that didn't have the heat, so the flavor. And what we ended up with is this pepper that I always I show this picture a lot, but this is a good example of being able to get out and really talk to growers and producers and consumers and retailers. Originally, we were going after trying to make a habanero pepper something that really looked like a habanero without the heat. And what we heard back was that no retailer would dare ever sell it because a regular habanero could hop in to not hop in, and if you couldn't tell them apart, that would end in lawsuits. So what we ended up doing is going back a few generations where it had these different elongated peppers, and actually this worked out well because they had a lot more aroma and flavor, and the yield was just incredible. So we started to do a breeding project for this habanero without heat, and then later on developed the tools to start to actually look at what was going on in more detail. So a lot of this makes use of some technology. I think is also now up at Geneva in the viticulture enology program. But instead of a regular gas chromatograph, what this one is doing is it has a polymer fiber that's able to sample all the aroma volatiles that are coming off of the sample. It runs a standard GC mass chromatograph run, but in addition to just the mass spec detector, the other detector is U. And so it's a matter of sniffing along as the machine is measuring all the different compounds that come out. And as you have a different sensory reaction, oh, this smells grassy, or this smells catty, or this smells fruity, you can take those time points and those percepts and match them up with the MS results. And so we were able to get some hits where we could line up a chemical like beta-ionone with a percept, but in general, there's a disagreement between how abundant a compound was that we could see with a mass spectrometer and whether or not we actually were smelling it or sensing it. So, for instance, a lot of these esters are quite abundant, but they're really not abundant enough to be able to smell them in the GCO, probably mostly because of the threshold of the compounds. But the most interesting result of this was the finding that all these aroma volatiles and everything that smelled good about a habanero pepper was coming from the same part of the pepper that's really hot and spicy. So on the inside of a habanero pepper especially, these white ribs that on a bell pepper is what you would throw out while you're cooking it, this is covered with these yellow glandular blisters where the capsaicin comes oozing out in great quantities. And in that same oil, you find all the aroma volatiles. And so here it shows the GCMS profile of a whole habanero pepper, and here it's M count, so millions of counts. And every other tissue of the pepper was really didn't have any of the aroma volatiles by the mass spectrometer or to smell them. So here's just thousands of counts of just seeing the baseline here. But if we were to just cut out this placental decipiment, the white ribs here, and put them in the machine, then we get back up to millions of counts in a similar profile before and all the aroma. We can even take a Q-tip swab and just wipe up some of the oil. And then that, of course, was a very spicy Q-tip, but also that had lots of all the aroma volatiles back. So the aroma volatiles are coming from the same tissue as the spice. And so it's been a question of then, okay, so how can we get hot pepper flavor without the heat? Can we do it naturally in a pepper? And with the one habanada pepper, the habanero without the heat, we had mostly done that. And so it was a question of, so how does that work? And so it was actually just a matter of using some different alleles that were different mutations that result in the lack of heat like you'd find in a bell pepper. Some different alleles from different species can decouple the heat production from the aroma production. So our collaborator in New Mexico, Mary O'Connell, had been doing some very nice SEM micrographs to start to look at the blisters and these oil-filled blisters. You can see the bumps here on this tissue. And then comparing that to the much smoother, unblistered skin you'd find in a pepper that's not hot. So it's lacking these capsaicinoid blisters. So with the discovery of the CT scanner next door, we, of course, ran right over with some habaneros, and Mark was game. So in Geneva, I'm going to play the pepper movie now. And this is just one of my favorite runs we've done. And so here is, again, the 3D rendering from the CT scanner, letting us actually start to look inside the peppers. And one of the great things, and I just love these photos because you can see all of the blisters all along the inside of the pepper, and actually start to count and account for area. And when we started to do some of these scans, one of the reasons we were doing them is just because it was fun. You know, it let us see some different perspectives on what we were working on in the lab, and definitely, you know, as I talk about some different plant tissues in class stuff, it definitely helps me engage as an instructor better. But it's just, for us, it's been remarkable, and you can see the detail in here, just being able to see all the different blisters. What actually, after looking at some of these, we started to make some observations that we need to now follow up. It's been just really exciting. This is a habanero pepper. It used to be the hottest pepper in the world. It's not the hottest pepper in the world any longer. There's this new pepper, the boot jalokia or the naga jalokia. So these new super hot peppers that are 3 times hotter than a habanero. And so one of our questions has been, as geneticists, is can we break it down? So what are the genetics that allow it to be so hot? One of the tricks we already know, I think we know, is that they measure heat in the pungency or the amount of these capsaicinoid alkaloid compounds per gram of pepper. So you don't necessarily have to produce more capsaicinoids, you have to produce more heat compounds, you can just produce less grams of pepper. And looking at the peppers, one of the interesting things you find is, so here are these placental ribs, which are naturally very light and very pithy after doing lots of RNA extractions from lots of habaneros. You start to notice things. So one of the things we notice after dissecting several hundred habaneros, it's something to become ingrained in you, is this tissue is very light, pithy, fluffy. Those boot jalokia peppers that are much hotter than a habanero are also, their whole pericarp, instead of being a heavy, fleshy pericarp, is also light and pithy, like the placenta. So one of the next things I'm going to do is grow some more boot jalokias and take them over to Mark, because it will be very interesting to be able to get some scans of those and really to compare the densities, and that might be the way we start to quantify some of the phenotypes, at least in some of the parents, to see, is this really what we're seeing? And so again, in the mild habanero, the secret has really been starting to take advantage of some of these other alleles that have other mutations instead of the major deletion. You find in a bell pepper, there is just a simple indel frameshift mutation in the chinense background that we've been breeding with. And the original experiment we did was just to cross a pepper that had this mutation, so it wasn't hot, it didn't have any aroma, with a habanero that had all these aroma peaks. And then in the F2 population, a nice, simple relationship showed up where we have a straightforward relationship where we have 2 groups of peppers, so we can do a principal component analysis of the aroma. What basically gets separating out is the presence of these big ester peaks here. But we have a group that contains a lot of the aroma, the wild-type volatile profile, capsaicinoids, and is wild-type for this gene. And so we have the one gene, and that's what's controlling whether the pepper is hot, and that's controlling whether or not these aroma volatiles are produced. So it's basically just one big switch turning both pathways off and on. But we are able to get some peppers that start to have this aroma back. And so here, and this is the old way of looking at the capsaicinoid blisters. We have these large yellow blisters on a wild-type plant. We have the lack of those blisters on the mutant habaneros. And on the leaky alleles, it literally is leaky, in that some of the mutant plants, we get a little blister with a little bit of that spicy oil coming out, and also quite a bit of the aroma coming out. And so this was really exciting to us as it started to explain actually what's going on in the pepper's metabolism. And basically we know now we can kind of turn off and on heat and aroma and know they're coupled. Another opportunity that's come up though is now is that we know more of this, there are also other peppers that are hot but lack aroma or have the aroma. And now we're starting to move towards some of those populations where we have the heat fixed but aroma is segregated. And you can see just the really intimate relationship of the aroma volatiles and capsaicinoids once you go back to the biosynthetic pathway. And what, again, we're going to get back to the phenylpropanoids again, where you have a typical pathway that's found in a lot of fruits to produce aromatic aroma volatiles, where you have an alcohol added to an acid to make an ester, and then it's just a matter of, in the capsaicinoid molecules that's going to make the pepper spicy, it's many of the same precursors for the fatty acids can be made into the carboxyl tails that form the tail of the capsaicinoids. And so we have two metabolic pathways that are sharing some of the same precursors. And actually it does look like they're competing for some of the precursors. Because the most common branched groups that are incorporated into the tails of the capsaicinoids are the least common that's available in the same glandular areas for the aroma volatiles. So those were the three stories I wanted to share about some of the different quality aspects we're looking at and how it all kind of centers on the phenylpropanoid pathway. And it's work that we are just gearing up on, but I wanted to share kind of where we're at now and I think the directions you can see for the future. And I want to thank a lot of people that contributed to this. Chris Smart, Amara Dunn, and lots of people from Extension. We're working on some of the Phytophthora disease resistance. The high tunnel work in collaboration with Chris Ween, and a lot of help from Leah, Jessica, Liza, and Michael. A lot of the Phytophthora breeding for the peppers was originally done by Matt Felice here. The anthocyanin work I showed was done by James Keach Ryko, and Jim Myers is helping us with a lot of the breeding work on the stringless snaps. The seed work was done by Lindsay and Nancy Iannetta. The images, Mark over across the road. Nancy Iannetta did a lot of the work on getting some of the markers up and running in some of the new fluorescent SSRs, and David Bollier at CalSec was instrumental in getting a lot of the aroma analysis done. And thank you for the opportunity to come here and share this with you today. Yes, Chris.
Michael, I was wondering, as you take the husk off the squash or pumpkin seed, what happens to the germinability and its resistance to soil-borne organisms and germination in cold soils and stuff?
It becomes an issue. You know, it's kind of the counterpart to the pea story that way, where in pea we're interested in how can we get a more durable seed coat that's more resistant, and here this is by virtue of what we're doing, we're trying to remove that. The main issue we tend to have is actually viviparity of the seeds. If we don't get them out of the fruit in time, they do tend to germinate. So it's been a learning experience of how to deal with that, and I talk to some growers and seed producers that actually, you know, they do this quite well. And in Austria, it's one of the national crops, so I think they have it down well. It's been a matter of learning some of their tricks. Yeah, yeah.
I've done some work with apples where we're looking at polyphenolics, which is very much a genotype thing, but there's a big environmental difference too. If we look at one genotype over 3 or 4 years, sometimes we'll see an order of magnitude difference between one growing season and another. Do you see that with the hot peppers as well, or is it more consistent? Yeah,
and I think it's in the peas in the flavor. That's why the bitterness will come and go. It'll be a similar thing. With hot peppers, it's definitely an environmentally responsive pathway. You know, it's also a matter of rain. If you have a hot, dry summer, you will have some hot peppers. I'm really also interested. We've never really. There tends to be lots of inherent variability even between fruit on a single pepper plant as to how hot they are. It's always been a difficult experiment to do. But a lot of the type of work Andre Kessler does, looking at how you can have a plant cue to another plant, is the work they do with you know, methylgesminate inducing the alkaloids in tobacco plants. could also probably have a lot of relevance in a hot pepper.
So one thing I've always wondered about that, is it a question of genes that are turned on and off on environmental cues, or is it a question of substrate limitation or more quantitative differences between one season and another?
I don't know. There's been a lot of studies on that, but they all seem to have differing results. It's been a matter of, again, even within the pepper plant, there's so much variability. So how can you get a very accurate measurement? And also, people have tended to do different studies with their own favorite type of pepper. And so there could be just, I think there are some peppers that are much more environmentally responsive than others, so they naturally just have larger error bars than others. But I think it's, no one knows. I think it's because it's been really hard to actually quantify how hot a pepper is, and even for a lot of the hot pepper industry, getting a pepper that's consistently hot is a big problem. And often what they'll do is have the not hot blend, and then just add some hot to it, so they can actually calibrate the amount of heat by adding it rather than trying to just achieve it through the harvest.
For the diffuse seed oil study, do you know what the dark colors are like what pigments they're attributed?
Yeah, so it is the pre-chlorophyll, what's the? Protochlorophyll. And then also lutein is quite abundant.
So if you make oils out of it, doesn't it cause oxidation when you have abundance of chlorophyll in it? Because I know that in canola industry, when you're working with canola oil,
right.
They want to separate out all the green seeds so that it doesn't cause oxidation in the oil.
Right.
So if it has such high abundance that it stains white clothes, wouldn't have like an oxidation effect?
Well, is it also a matter of the, so there with the green seeds, is it a matter of maturity or?
Well, they use it, they know that chlorophyll presence in seed is attributed to maturity of the seed. Like a less mature seed has more chlorophyll, and a mature seed has less amount of chlorophyll. So you can use that as a maturity index. But I think in their case, it's more about when the weather is cold, the seeds don't mature fully by the time they harvest. So that's why they have green seeds. Yeah. So then that can cause the green color pigments leaching into the oil, which then causes for like quality damage.
Yeah. So I think the difference might be that these seeds are fully mature. I know with some of the other oilseed crops, there's a big interest in black versus yellow seeded brassica naphis, and so there it's an issue of with the black seed, you have a stained oil, and you get less oil yield. So here I think it's definite. I don't think there are many issues with the stability of it. It tends to be a cold pressed oil, and so I think there are, to retain the maximum health benefits, usually you use it before too long, but I don't think there's any major oxidation issues with it. I don't know why. That's a good question.
50-some years ago, I did my PhD thesis on A gene in peas, and I was interested in tasting the bitterness in the seed coats. And my advisor, Bill Kelly, was able to taste it very readily, but most people could not taste it. Have you? You've talked about the genetic variability as far as the crop is concerned, or season is concerned. What about people? Do you find differences there?
So far, I was very proud to show lots of people my purple peas, and kind of universally they said, this is really nice, but it tastes a little off, Michael. So I think so far, and so there we're looking at snap peas or snow peas. So we're actually looking at the pod, and the seeds are small and only partially developed.
Right.
So I haven't done much look at the seed coat, or actually taste the seeds as much, as we tend to be more interested in the pod, but we will probably be finding that out. But I think it's, and it's a flavor that it's not an objectionable flavor, it's just not something you would normally expect to find in a really sweet, succulent snap pea.
Well, Kelly would tell me that in the seed coat of the pea, it was astringent.
Mm-hmm.
Yeah.
And that's definitely what everything says. And there's a single gene, palins, that would then remove that. And so it hasn't been studied much in quite a while, to my knowledge. But I think that's.
I found that there was a gene MP and another one in K, and I don't know if you know Markerassisted. There's a Swedish breeder that had. I had a line of his, line 578.
Mm-hmm.
It had 5 recessive genes. So even though A was dominant, it was free from and not only any anthocyanin, but any of the, what we call the metabolites anthocyanin.
Right, yeah, the upstream. Yeah, no, there's definitely, pea is, even though I love genetics, pea is one of the areas where I definitely feel a little intimidated, because some of the genotypes for some of the lines we're working with will go across the screen a couple times. There's a lot in there, and I think it's still the key and the fun is looking at all the epistatic interactions between them. And the aging was just recently cloned and elucidated, so they now know the transcription factor that causes the anthocyanin upregulation. I think there's going to be a lot of potential in the near future to then find all the other transcription factors that can turn on anthocyanins and all the other parts of the plant. Another 2 transcription factors further to go from just having a purple flower to a purple pod.
The other thing, have you tried the B gene with your snap peas? It's rose-colored flowers. There's cyanidin in the flower. I think in the flower, but certainly in the seed coat, it's instead of delphinidin, leucodelphinidin was leucocyanin.
Mm-hmm. And so, and that's actually what is in this slide here is. I thought I had the great idea of reading these papers where they had these red potted peas. And I was like, well, this would be fantastic. And then a little bit later, I find on the internet this woman that's doing it in her backyard quite well. Yeah. So it's and so that is a gene that I think, especially that's interesting to me because my neighbor in Bradfield was Walter DeYoung, and so he was. It's really similar to what goes on in potatoes. So it's probably the same gene, and it's just a matter of cloning it and starting to study it. One of the big interesting things for us has been how you have different diversities of anthocyanins in different tissue types. So you have a transcription factor to turn on the presence of anthocyanins, but you don't always get the same anthocyanins all throughout the pea. So it'll be another interesting thing to learn more about. If you have things to share, I would love to find out more.
I'd like to talk to you.
Great. Should ask if there's any questions from Geneva?
Yes, no.
Okay, thanks. Okay. Thank you.
This has been a production of Cornell University. On the web at cornell.edu.